So today I'm going to talk to you guys about coral reefs. And, my research has taken me to a number of different areas. I've done work on predator-prey interactions and the effect of micro-pollutants, plastic micro-pollutants in the oceans. But where I'm mostly focused on are coral reefs. And here's this image over here of these tropical areas. You can see here's a tropical island. You can see the reef forming around it. Here's part of an atoll. You can see this reef over there. And so the question is, what exactly is going on that reef? And what I'm going to do today is, I don't know how much prior information some of you have or don't have. So I'm going to give you a background on corals in general. And then from there, we move on to some of the problems and issues that the corals are dealing with. And then we'll follow that up with my own research and what I'm working on as far as the coral reefs are concerned. And then we'll wrap it up. So those same images that you saw from above, the aerial pictures, if you lift the sort of the veil of the water and you go under, this is what you see, right? Those beautiful, amazing, magical animals over there, the organisms there, known as the coral reefs. Now, you might sort of be thinking, why do we care? Who cares about the coral reefs? They're pretty and all, and make for great diving and so on. But why do we care about them? They're actually fundamentally very important, right? About 25% of all marine life, either directly or indirectly, relies on the coral reefs, okay? So the coral reefs cover less than 1% of the ocean floor, but about 25% of all marine life relies on the coral reefs. Coral reefs are also important not only in tourism and recreation, but also as far as food and fishing is concerned. They provide a source of protein to over 1 billion people around the earth. There's potential to get medicines from the reefs. And of course, areas where the coral reef is healthy, it protects those areas from storms and so on. So fundamentally, they're really important. And for those of you interested in climate change and so on, the coral reefs have been this great sink for carbon dioxide by absorbing the carbon dioxide as they grow and sort of maintain their skeletal systems, so that as the coral reefs, if they get not only damaged, if they start going extinct, it's going to have implications and ramifications all around the world. So here on the top, I've got a couple of pictures of corals. This is what you see when you go diving on the reef. And initially, of course, they look like rocks. They look like stones. They don't even necessarily look like animals. But in the lower panels, you will see, I'm taking a closer look, and you can see the individual tentacles, right? You can see the individual tentacles and polyps of these organisms. And that's because they are, in fact, animals, right? As a review, I'm sure most of you have had your classes in, about animals and so on, and have covered classifications. But as a reminder, these guys are within kingdom Animalia, within phylum Cnidaria. And again, as a reminder, within phylum Cnidaria, we've got things like cubozoans, which are the box jellies, scyphozoans, which are the jellyfishes, the hydrozoans, which are things like the Portuguese man of war and hydra and so on. And then we've got this group called anthozoa. And these include everything from sea anemones to zoanthids to gorgonians, and of course, the corals. And when you go diving, this is what you see. And these we're now referring to as hermatypic corals. These are reef-building corals that contain symbiotic dinoflagellates. These are algae that live within the tissues of the coral. And these corals, as they live and grow, and as you can see in this picture, they end up becoming the basis for these clear, shallow water tropical ecosystems. Okay, so in these areas, the organisms either feed on corals, directly or indirectly, or feed on things that feed on corals, or use the coral structure as a place to live in or to grow their offspring in and so on. Now, these are tropical organisms. You can see in this diagram, the pink shaded area is where you find most of the coral reef communities. And the dashed sort of pink line, north and south, that represents the 20 degrees Celsius isotherm. So in between these two lines, the temperature doesn't drop below 20 degrees Celsius. These are warm water organisms. There are a few exceptions over here towards Taiwan and Okinawa and the northwesternmost Hawaiian Islands and Bermuda. But in general, these are found in the tropical warm waters. But of course, not too warm because that becomes problematic. And here's another image, some sort of fun fact. Here's the typical tropical images, and I'm sure some of you are aware of. Some of you have spent time in the tropics, have seen these beautiful white sandy beaches, and even those white sandy beaches come from corals. Here's a picture of a parrotfish feeding on the coral, and as you can imagine, this exactly here is parrotfish poop, and that ends up becoming those beautiful sandy beaches that you guys like to go and hang out and play in. All right, now back to the science again. Now these reef building corals come in a lot of different forms and shapes. And I put here and here, I say they all contain symbiotic dinoflagellates. And I put the all in quotations because I acknowledge there's a couple of exceptions to this rule. But in general, almost all of them have symbiotic dinoflagellates. And some of you may have heard the term zooxanthellae as the name of the symbiotic dinoflagellate that lives amongst corals and other invertebrates. And here I'm putting the term zooxanthellae under quotations again, because the term zooxanthellae was created when we thought there was this one algae that is symbiotic with all these different invertebrates. We thought there was this amazing algae that could do this. We have now realized that it's not one algae, it's actually hundreds of species of algae. And so we put them all within this family Symbiodiniaceae. And it's this very relationship between the animal, the coral animal, and this algae that allows the corals not only to survive, but actually prosper in what are otherwise nutrient-poor waters. Okay, in the picture of the tentacle, you can see those tiny little circles. Those are the algae that pack the coral tissue. And so these tropical waters, again, they're very nutrient poor, right? That's part of the reason why they're so clear. So not a lot can grow in there, but corals, because of the symbiotic relationship, are able to do this. And it's a very interesting relationship, right? Here, this green circle here represents the algae, the dinoflagellate, and here's the animal host, the coral tissue, right? The algae, of course, can photosynthesize, can make its own food, and that's really powerful. And as it makes its food, it takes whatever food it needs, but then gives the rest of the photosynthetic material to the animal. So the animal part, the coral part, can potentially get upwards of 98% of its daily caloric needs under ideal conditions from the algae. Well, that's great for the coral. What does the algae get out of it? Well, a few things. The algae gets micronutrients. Now, the coral, in addition to having the algae, in addition to having these dinoflagellates, it is still eating plankton. And as it eats plankton, it has waste product. It has nitrogenous waste. And it's that very nitrogenous waste that's the micronutrients that the algae need. So the coral gives the algae those micronutrients. And the algae gets not only those micronutrients, but also within the symbiosome, within this vacuole where it lives inside the coral, it gets the perfect growing and photosynthetic environment. This diagram says pH less than 6. It actually is less than 4 inside that vacuole, and that's a perfect environment for these guys to very efficiently photosynthesize. So they're really helping one another, right? They're both better off. And that's why we refer to this as a mutualistic symbiosis. And again, I have to emphasize, we're talking about dinoflagellates. These are single-celled algae with the two flagella, perfectly capable and happy to live within the water column. In fact, around the reefs, when we take samples of the water, we find plenty of these dinoflagellates. But when exposed to animal tissue, they drop the flagella, they metamorphosize in a circular form, and are capable to live within the coral host. So the coral doesn't digest them, it takes care of them, and they create this mutual relationship. And again, I can't emphasize, these are algae. These are organisms living inside the tissue of another organism. It's a true example of endosymbiosis. These are pictures that my colleagues and I took a few years ago where we're looking at the individual cells of these corals. And here, for example, you can see it's an individual coral cell with 2 algae inside it. Here's another coral cell with 4 algae inside it. These are scanning electron microscopic pictures of them. Here's a TEM pictures. You can see again, here's the algae and here's the host surrounding it. All right, so they live there, they're helping each other out and so on. Here's another picture. You can see clearly the algae living within the tentacles. And if you cut the tentacle across, here's a picture I took where I put UV light on the tentacles. And when you put UV light, the chlorophyll sort of fluoresces orange or yellow, and that's, you see the algae over there. And the animal tissue fluoresces green because corals have what's called green fluorescent protein. And you can see here are the algae packing the endodermal tissue of the host. Excuse me. And how do they get the algae? Well, it depends. In some cases, the algae are sent by the parental generation to the larvae. It's called a closed or vertical system. Or they are captured from the water column, and that's referred to as the an open system or horizontal system with where the original polyp is aposymbiotic. It doesn't have any algae in it, but then it gets infected and picks up the algae and the color. All right. Now, the symbiotic system is great and all, and it's doing wonderful, but under stressful conditions, the symbiosis breaks apart. And so I won't spend too much time on this. I'm going to very quickly go over a few of these slides because I know you guys have covered climate change and global warming. Just as a reminder here, here's a figure showing you how the temperatures are rising over time. And here's another sort of diagram showing you how the oceans are becoming more acidic, right? So the pH is dropping. So these are the 2 big problems. And it's particularly a bigger problem within the oceans. About 90% of the warming that's happened between 1971 and 2010 happened within the oceans. And this heat is already stored in the water, and as eventually it's going to come out, that's going to be very problematic. And of course, the causes are many. You know, the biggest cause, of course, is carbon dioxide levels. These are atmospheric carbon dioxide levels that we have estimated for the last 800,000 years. And we can see the highest level previously we hit was about 350,000 years or so, was 300 parts per million. Right now, the average for 2019 in this diagram says it's 409.8, right? That's a huge number. And what comes along with this high carbon dioxide level is the warming that we see. Here's another diagram showing the up and down movement of the carbon dioxide, but the general pattern is upwards. And I just looked this up a few days ago. We hit the peak for 2021. It was April 30th, so that's 6 days ago in Hawaii on the top of the Mauna Loa Observatory. It was 420.54 parts per million. And finally here, I'm sort of going over the fact that it's not just carbon dioxide, it's also methane, nitrous oxide, chlorofluorocarbons, and so on. They're all important, but again, the main players here are carbon dioxide and methane. And as these accumulate and the temperatures go up, they affect the reefs, they affect the corals. These corals that should look beautiful and amazing like this end up not looking so great. Now, there's a lot of factors that can hurt the corals, right? Everything from eutrophication and runoff to diseases, crown of thorns sea stars, ocean acidification, overfishing, storms, chemical pollutants, including but not limited to sunscreen, in addition to temperature. And your inclination might be to say, wait a minute, if the corals are so sensitive, then maybe they're on their way out anyway. But the fact is, these are organisms that have survived hundreds of millions of years. And just in the last 20, 25 years, because of human activity, they're starting to die off. And that is the problem. And under these stressful conditions, what we're seeing is happening is what we refer to as coral bleaching. What this is that as the coral loses those dinoflagellates, it loses the symbionts, it goes white. And it's not that it's turning white. The coral skeleton itself is white. And so when it loses the algae, the sort of translucent tissue allows us to see the white skeleton underneath, and we see this whitening process that we refer to as coral bleaching. And if this coral bleaching goes on a long period of time, what will end up happening is that the coral will eventually die, and the ecosystem can potentially collapse. Now, the research that I'm doing looks at things in a few different places. I have spent some time in Taiwan, in southern Taiwan, around Nanwan base. So some of the work I show you guys will be from this part. Some of the work I show you will be from here, Kaneohe Bay. This is the Hawaii Institute of Marine Biology. And some of the work that I'll show you will be from Moorea, which is in French Polynesia. It's the island right next to Tahiti. UC Berkeley has a lab here, and we do some of our work here. And before I move on, I just sort of have to acknowledge some of my students. These are some of the students that come to the field with me and do the work. It's great for me to sort of come up with the projects and stuff, but these are the individuals who actually do all the hard work. And the work that we do, here's another picture of some of my colleagues and students. And the work that we do is, again, based on what do we expect is going to happen over the next 50 to 100 years. And for that, we rely on various scenarios that models have predicted, right? So we have models that, based on consensus from thousands of papers, have sort of the IPCC has come up with. And what they've come up with are various possible scenarios. For my studies, we don't necessarily pick the absolute worst case scenario, and we don't necessarily pick the best case scenario either. We're sort of being realistic, and we pick the mid-range, most likely scenarios of the kind of temperatures that we're going to see, the kind of pH that we're going to see, and try to figure out what might be happening to the corals under those circumstances. So we set up various aquaria like this, and within those aquaria, we very closely monitor and control the conditions. We have control conditions representing conditions that we have right now, and then we have subsequent conditions that are going to mimic what we're going to get by the end of the century. And what we're realizing, of course, is under higher temperatures, we're getting this bleaching process. You expected that. And in fact, what we're realizing is that as this bleaching process happens, it's not just the algae that are being lost, but rather the entire endodermal tissue, that inner lining of the corals and these anemones are just sloughing off and are being spat out, right? So here's this healthy individual here. Here you can see these black dots represents all that tissue, animal tissue, as well as the algae that have spat out. And within a few weeks, you get completely aposymbiotic individuals that have lost all their algae. And so whereas the corals we want to see end up are this, we instead end up seeing images like these. These are not my images. I have borrowed these, I've taken it from some others, but you can see they look beautiful, but in fact, this whitening is mass bleaching of the corals. Here's another before and after image. You can see how the corals have bleached in the right panel. And again, you can see the effects can be really quick and devastating. Here, December 2014, here's a healthy coral. By February 2015, within a couple of months, completely bleached. These are whitened corals. They're still alive. If conditions were to improve, they could recover really quickly. But if conditions don't improve, they will be dead soon thereafter. In this case, all these corals were dead by August of 2015. The diagram I have here is an experiment I did showing that a completely bleached coral, potentially under ideal environments, could completely go back to normal within 4 weeks, but that's only if the conditions improve. So the question then becomes, as we look at these bleaching effects, is that, wait a minute, not all of the corals bleach. Some of them seem to be okay. So what is the difference? What's happening to the corals that are not bleaching? And what are the effects of temperature versus CO2 and so on? And one of the things that we consider, because not all the corals bleach the same way, is the potential for the symbiotic algae to be very different from one another. You may have heard the term symbiodinium in the past. We used to, again, think that even after we discovered that all of them were multiple different species, we used to still think that they're all one species. And we put them into various clades, clade A, B, C, D, E, F, G, and so on. We have since realized that each of these clades are actually a different genus of dinoflagellates, right? Symbiodinium, Breviolum, Cladocopium, and so on, right? And that each of those individual genus have subgenre, right? They have specific species and subspecies within them. Now, why would you have so many different species and subspecies of symbiotic dinoflagellates? Well, one explanation is that, well, between time and space, because of genetic drift, variation happens. Or it could be adaptive. It could have functionality. It could be that certain types of algae are better suited for different environments, right? Here are these sea anemones, for example, that we find off the coast of Southern California. It's Anthopleura sola. And you can see all the different color variation here is based on the type of symbiotic algae that lives within them. And studies have been done, and you don't have to worry about the details of this table. I'm just throwing this table out to show you that we have ranked the various types of algae. And we have seen that some of them are very resilient. They're very heat tolerant. In fact, they prefer warmer temperatures, and others do not. And so there is this general thought that corals that have some of these algae up here, those are the ones that are going to survive the higher temperatures, whereas the corals that have the algae down here are going to bleach and die off. Well, that's one possibility. And so my research, when we went to Taiwan, we wanted to look at a few things. We wanted to see what was the effect of the temperature, what was the effect of the lowering pH by itself, what was the combined effect of temperature and lowering pH, and whether one or more of these were influenced by the type of symbiodinium, or by the type of algae that was present in the corals, and whether this gave them heat tolerance or not. So here's Seriatopora and Taiwan, and this is the one main finding that we had. We looked at symbiont per centimeter squared, right? We looked to see what the density of the symbionts are. Under normal conditions, they were pretty high. And then we look at before and after. This is high temperature, ambient CO2 in the second panel here. This means under high temperature but normal pH, there was definitely bleaching, right? The population dropped, you can see here. At ambient temperature, high CO2, right? Under lower pH, but regular temperature, no bleaching at all. High temperature, high CO2, so high temperature, lower pH, again, massive bleaching. And there was no difference under the two high temperature scenarios, regardless of whether the pH was lower or not. So this first panel, the data showed us that, well, at least under the kind of light conditions that we get at 10 meters underwater, and at least for this species, it did not seem that the lower pH was causing bleaching. It was the temperature. Now, this is not to say that the pH had no effect. As we start looking at other things, right? We start looking at the chlorophyll per symbiont. We looked at chlorophyll A to chlorophyll C2 ratios. We looked at the division rates of the algae and so on. And here we start seeing that there was some effect of the pH. So there is some stuff happening with the pH. It's not to say that the lower pH is fine. And certainly the lower pH is affecting the skeleton of the corals. But at least for the bleaching part, it did not seem to be a factor. So it was the temperature. So then we go back to the question, did the algae make a difference? And so we looked to see at the symbionts under each of those different conditions, right? Ambient temperature, ambient CO2, ambient temperature, high CO2, high temperature, ambient CO2, and high temperature, high CO2. And we found absolutely no difference in the types of symbiotinia, the types of symbionts that we had, and the level or amount of bleaching. So it seemed, at least here, that this was not the big, that the algae was not the factor. So if it's not the algae, who could it be? It's most likely the animal host. And where my interest took me next was scenes like this when I would see diving, right? I would go on the reef and you would see a bleached coral reef with this one coral right next to the rest of them, perfectly unbleached. Species that looked identical, one of them bleached, the other unbleached. So the question was, what exactly is going on here? So the next research, we went to French Polynesia, and this time we worked on Poslopra meandrina. And here are some of these Poslopra meandrina we collected from different parts of the island. We set them up in these water tables for them to recover as we fractured them and put them into plates like this. Here's one of my students. We had to go and clean the corals on a daily basis. And here's my other assistant. This pufferfish came and hung out with us every day that we went to work on the corals. Anyway, we took these corals and then we decided, all right, let's see how these corals respond under different conditions, different temperature conditions. And we looked at them at 25 degrees Celsius, 27.5 degrees Celsius, and 30 degrees Celsius. And you can see a lot of variation from the error bars here. In fact, statistically speaking, there isn't much happening here, maybe some bleaching here, maybe not. It was difficult to tell what is going on. But the question this time was not whether these higher temperatures are causing bleaching or not. We had gone to French Polynesia because we had discovered that the corals there seemed to be resilient and heat tolerant. So what we started doing was, instead of looking at the groups of the corals, we started looking at the individual coral colonies. And here something very interesting came up. What we see is that under each temperature condition, we have individual coral colonies that are reacting very differently to the temperature profiles. Here at 30 degrees Celsius, which is a very warm temperature, we see the individual coral colonies that are very quickly losing algae, some that seem to be, just holding on, and a few that seem to actually be doing really well in those higher temperatures. And this is very interesting. We noticed the same thing when we looked at the effect of temperature on change in chlorophyll per surface area. Not much happening here when you look at the whole group, but again, individually, big differences, right? Individuals that seem to do just fine at higher temperatures and those that do not. And this is really fascinating, right? Because whereas evolution is happening at the population level, whereas we look at bleaching versus non-bleaching at the population level, natural selection and potential for survival happens at the individual level. So it becomes important to consider what's special about these individuals that seem to do just fine in warmer temperatures. Now, here's a different location and different species of corals. So once again, we looked at the different symbionts, right? And here I'll show you this. I haven't done the graph yet, but we found that a huge slew of different types of algae associated with these corals, right? Each of these red ones, C1, C42.2, C1b, C3, and so on. A lot of different types of symbionts. But no matter what we realized was that there was no correlation between the type of algae that there was, the type of symbiont we had, and the response. Once again, we come back to the fact that whatever is happening, it seems to happening from the animal side, from the coral side. And as we speak, we're still working on it. We haven't finished the work. My collaborators are looking at the animal tissue of these samples, and we're looking at transcriptomics. We're looking at the RNA from these tissues, trying to figure out what is different between these animals and how we might be able to use this to explain the variance within the individual coral colonies. So to what end? Well, the point is that if we can find these corals, the characteristics that allow them to survive versus those that don't, well, we can model what's going to happen by the end of century better. We can sort of certainly do reef management in a better way. But also there's a lot of people out there, what they're doing is they're working on coral assisted evolution, right? So they're looking at, let me see, I don't know if you can see this. They're looking at bioremediations and assisted coral evolution, with the idea being to creating corals, super corals, if you will, that are able to withstand the conditions we're going to be able to have by the end of the century, and hopefully be able to use those to reseed some of the reefs to hopefully end up with scenarios like this, where the corals are back to being healthy and doing well. So take-home messages here are that the reefs are threatened but not doomed. Climate change, pollution, overfishing are all major issues that need to be addressed. Local proactive intervention, all that stuff is great. It builds resilience, but reefs cannot be climate proofed, right? Prevention is great, recovery is possible, but no matter what we do, unless we address the climate change, unless we reduce carbon dioxide and other greenhouse gas emissions, the reefs will not be able to survive. So it's sort of like we can do the local short-term things to buy time, to get resilience, to be okay in the short term, but for the long-term success, we do need to eventually address the greenhouse gases and climate change. And I'm going to wrap up the part of my talk about the corals themselves. And I'm going to spend about 5-6 minutes for a quick sort of call to action. You know, in the past, as scientists, we used to just sort of say, hey, I'm a marine biologist. Look at this picture. 71% of the ocean the world is covered by water. Isn't that wonderful? I do my research. And then we would publish our articles in scientific journals, and we felt that we were done with it. But with everything that's going on in the world, we find ourselves in a position where we need to take action. And so that's my call to you guys, to do something about the issues that we're having. For me, it's the coral reefs. For you, whatever it might be, right? When it comes to the oceans, there's this tendency to think that they're so big and they'll be fine. These couple of diagrams really show you how much water and atmosphere we have, right? Compared to the size of the Earth, all the ocean water is this tiny little circle here, right? This tiny little circle represents all the fresh water we have. This pink circle represents the atmosphere in the air. It's not that much. We can affect it, we can influence it. And we have this image of the beautiful open oceans and wonderful and dolphins jumping out of the waves and stuff. But of course, you know that there's a lot of issues and problems in our oceans. And I get it. It's depressing, right? It's depressing, and it's okay to get a little sad about it. Honestly, every time I go to environmental talks, I come out of it so depressed. I'm like, well, the world is coming to an end, right? Might as well go have a big party on our way out. But that's not the case. The fact is that we put up all this information to let you know how important the issues are and how critical it is to take action now, because there is still time, particularly the next 10, 12 years. And you guys right now watching this video or this lecture, it's your generation that's going to make the difference. In order to make the difference, you need to take a few things into consideration. One is the interconnectedness of the world to one another. I love this cartoon, right? We happen to be privileged living where we are, right? We're in this part of the earth where we have our privilege. We're not in the sinking part just yet. But the fact is the boat is one boat. We only have one earth. Anything that happens anywhere in the world affects all of us. The next thing, in addressing these issues and problems, it's easy to sort of want to focus on just the environmental sustainability. But the fact is that we need to recognize that environmental sustainability, the ecological health, social equity, and economic vitality go hand in hand. And it's that intersection where we have the solutions. And so I love a few of these quotes I'd like to put up from MLK Jr. He says, I have decided to stick with love. Hate is too great a burden to bear. And he says, the nature of all problems, and you have to really think about this, right? When you come up with problems, they're either conceptual or management. Forget about the management problems. It's the conceptual problems that we have to deal with. When it comes to conceptual problems, we want to sort of address them from a place of love, not fear. And it makes a difference. Your actions will make a difference. Right? Here's another quote I love. It says, it is a mistake to conceive of choice and decision-making as a process of selecting from existing options according to a fixed formula. That omits the most important element of decision-making, namely the creation of new options. Think outside the box and come up with the solutions. There's a lot of things that you can do at the individual level. I'm sure most of you have seen diagrams like this. The choices that you make do make a difference. Some choices have a bigger effect, like the food choices and transportation choices, the number of kids you have. Other choices make smaller effect, but they all make an effect, right? The sort of cheesy standard things, reducing your carbon footprint, thinking globally, acting locally, and this idea of, instead of recycling, rethinking, refusing, reducing, reusing, refurbishing, repairing, repurposing, or recycling, all of these can and will make a difference. And I want to finish with this one picture of the Earth from the moon. It's the one Earth that we have. Right now, we're using resources as if we had 5 of them, and we don't. We just have this one Earth. And the actions that you take will make a difference to this one Earth. So again, a couple of quotes and we'll wrap it up. One is from Margaret Mead saying, never doubt that a small group of thoughtful, committed individuals can change the world. Indeed, it is the only thing that ever has. And the other one, again, an MLK Jr. quote, it says, if you can't fly, then run. If you can't run, then walk. If you can't walk, then crawl. But whatever you do, you have to keep moving forward. Take action, whatever that action might be, right? Don't give up. If you're going down, let's go down fighting, right? But maybe, just maybe, we'll fix all of this. So let's see what we can do. And that's it. I'll answer any questions you might have.
Okay, that was the end of the lecture. If you guys have any questions, please send them to me in the chat, and then I will pass it on to the professor here. But I have a few that I can start with right now. All right, the first is that you mentioned that coral can recover from being bleached within 4 weeks, but under what kind of conditions would this be in? Is this if the water turned completely back to normal in the best conditions possible, or would that rapid recovery occur even if the water was slowly turning back to normal?
That's a very good question. The 4 weeks, first of all, with bleaching, you have to sort of say how bleached, right? Because there's different levels of bleaching. So, and again, of course, during the recovery, how ideal is the recovery period or the recovery conditions? So the one that I showed you where the recovery was in 4 weeks, that was a completely bleached, like worst case scenario, all the way to full recovery, but under absolute ideal conditions. Chances are you're not going to get absolute ideal conditions, but also chances are that you didn't go to completely bleached either. So it was a partial thing. So it's a difficult question to answer. I should also mention that one of the areas that I'm looking into is that, yes, I did mention that the type of algae does not seem to make a difference in the bleaching process. But I have a feeling that it might make a difference in the rate of recovery. So the type of algae, the type of symbionts that might be available in the environment might make a difference on how fast this recovery is. So I know it's not a straightforward answer, but again, recovery can happen from anywhere from a few weeks to a few months. It depends on the conditions, the species of the coral, the species of the algae, how bad the bleaching was, what else was going on at the time.
Yeah, that's very interesting though, that the algae can make a difference in the recovery period. The next question is similar to that first one, in that it's talking about how much coral reefs can be saved. And it says, since reversing the effects of climate change would likely take many years, is it estimated that the majority of coral reefs would die off by that point, or do you think that they can be saved without losing too much?
That's a big question, right? So it's difficult to answer. But here's the thing. Are the coral reefs going to be the way they were? No. We're past that point. The reefs, the way they were 30 years ago, 40 years ago, we are not going to have the reefs that way at the end of the century. The reefs will be different. The question is, will there be a effective, vibrant reef system with whatever species diversity or whatever types of species that we might think, will that be around by the end of the century? And there, I think, combination of us trying to address climate change to at least slow it down, combined with local activity of trying to reduce other factors that might be affecting the corals, to lower the rate of damage there, along with local proactive action, as well as things that we talked about, right? Bioremediation and use of supercorals and stuff. I think some combination of all of those efforts will allow us to have some form of reefs around the world by the end of the century, but it will inevitably probably be different than what we're seeing today.
Okay, the next question is again looking at how efforts could save coral reefs. And it says, I know you said that algae dying off isn't a major issue, but would transplanting algae into coral be a feasible solution for areas where some of the algae has died off, or should the efforts focus primarily on the coral itself?
I'm not quite sure the algae dying off, what we're talking about here.
Oh, yeah, I didn't
Which
quite
algae?
I didn't quite understand that question either. We'll just move on to the next one then.
I will, there's part of the question I think I might have understood, because there's a tendency there to think that you have the coral, right? Here's a coral. And if I have some of those symbionts and I come and sprinkle some of the symbionts on this, especially like the ones that are heat tolerant, that this coral will take it up and be okay. And if those types of algae were giving these corals some super ability to survive higher temperatures, well, that would be wonderful. Except that it doesn't seem like it works that way. It seems like the solution of the corals being able to survive and handle it comes from the coral. So if all we do is we take some corals and we grow them somewhere and bring them to the reef, if they're the type of corals that are going to bleach anyway, right? Well, if the temperatures are going up, if we're not addressing climate change, if we're coming up with the same types of algae, coral, with the same type of dinoflagellates that are not resilient, well, next summer they're dead anyway, right? So yes, there is place for bioremediation. It just has to happen based on the scientific knowledge and understanding and a systematic way that addresses things. There's a lot of people, there's groups that they mean well, but they're just like randomly growing corals and trying to take it to the reef and trying to be helpful. And it's wonderful, they're trying, but it's not, their actions are not necessarily science-based. And a lot of their efforts, like I see, I go diving and stuff like that, I see a bunch of like their replanted corals that are dead, right? It looked good for the first month after they did it, but you go there the year after year, they're not there anymore. So, there's some things that can be done as long as you do it the right way.
Yeah, I think that's what that question was trying to ask. Great answer. And then the final question that we have is just one from me. It's because many members of InterSTEM have expressed interest in conducting experiments of their own in research. What ways would you suggest that scientists and researchers could share their findings beyond the scientific community instead of just in journals?
Yeah, I mean, anyway you guys can figure it out, right? I mean, when it comes to information technology, I'll be honest, I mean, I think your generation does this better than anyone else, right? So you guys tell me, right? I know there are various websites and people just, anytime they have a question, they go Google it and so on and so forth. But honestly, when I do my work and I was like, okay, I need this word to get out, guess what? I tell my undergraduate students, the 19, 20-year-old students, all right, guys, take this, go out there, figure out a way to get this word out. Because my training has been to, you write it up, you publish it in scientific journals and let the scientific process do its thing and get reviewed and challenged and so on. And I go to scientific meetings and present. But again, that misses it out on this other sort of area of getting information out to the general public. I do my part because I teach a lot of students. I try to tell as many students about what's going on as much as possible. I offer to give talks like this whenever I can. But beyond that, I'm all ears. That's what you guys are really good at.
That was the last question. And just one final question, like on behalf of everybody. For people who are interested in this topic, how would you advise us to learn more about like coral reefs and just marine biology or marine chemistry in general?
Just read. But be careful of what you're reading. I think Google Scholars is a pretty good source for information. Start by looking at review articles. Take classes. I'm offering online classes at San Marco College. Sign up for an online class. You guys are in high school. I think, if I'm not mistaken, dual enrollment for high school students is free. You can take my marine biology class. So those are options. And yeah, feel free to email me. I'm happy to send you some review articles on corals or coral bleaching or whatever to give you information. Whatever. Ask.